So thanks everybody for being here. So today is the fifth presentation of this term, the last one before Christmas. So it's a great pleasure for me to introduce Professor Moulton. He is a professor of applied mathematics at the Mathematical Institute at Oxford. He's also a fellow in mathematics at Balliol College. Dr. Moulton is working many things in applied mathematician, as I mentioned, but he does mathematical modeling and uses computational analytical techniques. He wants to answer questions in biology. He's focused on the continuous scale. He's particularly interested in mechanical biology, physiology, growth, morphoelasticity, and many other things. Before being a professor at Oxford, he was a postdoc at Oxford. And before that, he was postdoc at University of Arizona, if I'm correct. And he's a PhD graduate from University of Delaware. And he worked with John Pelesco. More or less, he's author of a book on new trends in physics and mechanics of biological systems. And from my perspective, he's doing very cool work. So I'm eager to hear what he has to say about the morphomechanical basis of shield forms. So thanks a lot for being here.
It's great. Thank you very much for the invitation. So I'm going to put this in full screen mode, which I think means that I won't be able to see anybody else. I tried this, so do stop me if you have any questions, but I don't think I'll be able to see you without coming out of the full screen. Yes, so we're going to talk about some work on seashell form. We've been working on various aspects of seashells for quite a while now, and I don't know. I think it's a nice place to start is to think about sort of why, and a good starting point is to think how and to observe how pervasive seashells are in our culture and in our everyday lives. Now, of course, the seashell starts out as a home for an aquatic mollusk, a mobile home, but from there they end up on our beaches. We collect them. They have been used as instruments, as currency, as jewelry, as architectural features, even as bathroom fixtures. Now, I think probably the reason that they're so pervasive in our culture is, one, that they're very abundant, two, they're very durable, and mostly that they're very beautiful. And in fact, these three things are a large reason why they garner scientific interest. So on the scientific side, what's interesting to note is, so they come from mollusks. They're built by mollusks, and mollusks form the second largest phylum in the animal kingdom, something like 100,000 different species of shell-producing mollusks today. I use the word seashell, but of course some mollusks might be living on land. Your typical snails in your backyard also fit this category. But mollusks have been around for a very long time, about 500 million years. And so there's a huge abundance in the fossil record, which makes them very interesting. And that's partly because the shells are very strong, so they're very, very well preserved. Now, what I want to think about is how shells grow, how they form, and how they look differently. Why don't all shells just sort of look the same? And what's interesting from a sort of mechanical and morphogenesis point of view is that shell growth is a fairly simple process. So the way it works is you have the mollusk living inside the shell, and think of a snail, right? Just a soft, slimy creature. And it's building its own house one layer at a time. And it's always building it at the current opening of the shell, which is called the aperture. And so all the action is taking place really around this single edge. And what happens is you have a part of the mollusk, which is called the mantle, which is just a thin, soft tissue. And the mantle, sort of living on right inside the shell edge, comes out and sort of wraps around the current aperture and secretes a new layer. And so from a mechanical point of view, there's this interaction between a soft tissue and a rigid structure, which is the current shell opening. And of course, it's a sort of dynamic relationship because the soft creature is being deformed by the shell opening, but it's also creating the shell opening. And that process is effectively conserved across all mollusk species. And yet through that same process, we get a huge diversity of patterns. Well, on the one hand, essentially every shell you encounter will have the logarithmic spiral pattern living somewhere in it. But there's a huge diversity of the sort of the broad form of shells, and in particular, very few shells are perfectly smooth. And so one of the things we really are interested in is what are called ornamentations. And so these are some of the sort of features on top of the smoothness of the shell. So the ribs down here, these sharp spines, we have in some cases fractal-like structures or wavy ridges, as you see in the bottom left. There have been a lot of modeling, previous models of shells, because these date back to 1838. This quote from the Reverend Moseley, who was working on geometric description of shells. The work I'm interested in really can be traced back to the pioneering work of Darcy Thompson, who brought in the concept that biological form is truly a realm of physics, which wasn't really appreciated before then. Starting in the 60s, you got the rise of what's called theoretical morphology, which really became about characterizing the diversity of shapes, sort of geometrically or in terms of a small number of parameters. A lot of work from Meinhardt, Oster, and others on the color patterns of shells. And there have been really very good computer generation algorithms for shells since the early 90s. But what I think is kind of missing in this picture is mechanistic understanding of the shell form. And so our goal has been to produce a mathematical framework that can reproduce shells, but in a manner that is not just making a shape that looks like the shell, but in a way that informs us as to how and why the shell looks the way that it does. So a framework that's intrinsic to the actual process of growth. And to really take this idea of Darcy Thompson and put it in very quantitative mathematical terms. Can we understand shell form in terms of physical forces? And really, can we find simple rules based on the physics of the growth process and understand the patterns and the diversity of patterns? And then taking a step back, so a lot of this work is in collaboration with a paleontologist. And so, as I said, there's a lot of studies on the evolution of shells and mollusks. And well, mollusks have been around for 500 million years. And people study the evolution of mollusks via their shell, and they're an excellent case study for asking questions of how evolution functions, because you have this amazing, well-preserved fossil record. What I will focus on today is a particular class of shell, which is the ammonites. So ammonites were an incredibly dominant species. Ammonite is a group. There are many species of ammonite. This is a group of mollusks that ruled the seas for 335 million years, but they've been extinct for something like 65 million years. Nevertheless, they are incredibly abundant. They were very successful. They had a high evolution rate, so there's massive diversity. They can be found all over the world. In fact, if you go to the southern coast of England, you can find, with very little work, you can find traces of ammonites. So, which is quite remarkable, because something like the person is holding here, it's quite an extreme case. But I've done this myself. You just comb the beaches and they're everywhere. It's remarkable because you're finding something that has been extinct for 65 million years just lying on the beach. Because they were so abundant, they're useful in dating, so biochronology, dating rocks in different areas. They've been extinct, but they have a close relative, which is the nautilus. And the nautilus is sort of this canonical shell, which is, I'm sure, recognized by everyone. So I'm going to ask a few questions about ammonites, but I want to start out with just thinking about geometry of shells. And the way we want to describe shell structure from a purely kinematic point of view is as follows. As I said, the growth is happening entirely at the opening, the aperture. So we have a curve, which is the aperture at any given time. And suppose I define a vector field on that curve, where each vector at any given point describes how new shell is being secreted. So it's got a magnitude and a direction. So I put a vector field on the curve, and that defines the aperture at a small later time. And the key to that is that I want to think in terms of a system that's intrinsic to the mollusk and the mantle that's actually secreting this material. And so our first step is in writing this mathematically, I'm going to work in a local basis, which is I'm going to attach at each point around the aperture an orthonormal basis, which in this case is going to be the tangent, normal, and binormal vectors, which are, as you see pictured here. So one pointing along the direction of the aperture, one pointing normal, orthogonal to the tangent, and then one pointing orthogonal to that, the binormal. And I'm not going to go into all the mathematics here, but you can write this down using tools of differential geometry for curves. And the idea is that you write that vector field, which is this V, as in terms of that local basis. And so because my local basis is tangent, normal, and binormal directions, my key players here are descriptions of the growth, VT, VN, and VB, in these three orthogonal directions. And if you work through, so the idea is you can work through the differential geometry, and the idea is to understand how different rules for those orthogonal directions of secretion result in different shell forms. And conceptually, it's quite simple. We have three simple ingredients to make a shell, as in the basic shape of the shell, which is it can expand, which is growth in the normal direction. It can rotate, which is growth in the binormal direction, but with a gradient. So imagine one side is growing faster than the other side, and you get sort of this rotation or coiling. And growth in the tangent direction is a bit strange to think about, but you can kind of equate that with a twisting. So to give some very simple examples, if I have only a constant growth in the binormal direction and no growth in the tangent or normal directions, hopefully you can appreciate that you're just stacking material, and I start with a circle. All I do is I build a cylinder. If I have constant growth in tangent, normal, and binormal directions, you create a cone, and it's a sort of twisted cone because you have this constant twist in the tangent with the tangent growth. If you have a growth only in the binormal direction, but with a gradient, so the cosine of S really just means, S is the variable following the aperture. So cosine of S really just means that in say the X direction, there's a linear gradient. So the outside is always growing at a faster rate than the inside. You can work that out, and conceptually you can hopefully appreciate that that would create a torus. And if you take that rule and then add an expansion on top of that, you get something that becomes basically the sort of ammonite shape or the nautilus shape. And if you, on top of that, add a component in the tangent direction, so now you're adding this kind of twist component, you start to get these non-planar what are called helicospiral shapes. What's interesting there is we can actually learn quite a bit from constructing the shell in this way. So there's another way that I could produce that same shape, which is instead of having a, what happened here? Oh no. There we go. Instead of having a constant, trying to move this little note is blocking the screen. There we go. Yeah, so instead of having a constant twist, if you have zero twist but have a time dependence in the binormal growth, which is kind of like having the point that's secreting the most in the binormal direction, suppose that point changes in time and sort of rotates around the edge, you get the exact same surface, but what you find is the surface looks the same. In fact, it's not the same, and you can trace sort of where particular secretion points would have been, and if you compare that to shells that have that basic shape, what you see is that the idea of twist is the one that is consistent with the actual cell tracks that we would find on shells. And so that's an important component that's going to come up again. So to give you kind of a concept there, the VT, this growth in the tangent direction, you can think of this as meaning that the animal, the mollusk, is twisting inside its shell as the growth proceeds. Okay, so there's no mechanics in that. That was just to kind of give us the basic shell shape. Now, if we think about ammonites, so ammonites are cephalopods, and cephalopods are characterized by a symmetric or very nearly symmetric left-right, sorry, a symmetry in their body. So they have a left-right symmetry in their body. The coiling that you see in ammonite, so these are lots of different species of ammonite. They all kind of have this logarithmic spiral. They're all coiling. They're coiling actually on the ventral dorsal direction. So they're coiling, in fact, on their dorsal. The dorsal side is the short side, and the ventral side is the long side. But there's a symmetry there because the logarithmic spiral is planar, and so if you flip the shell over, I have one here. I don't know if you can still see me on camera. Here's an ammonite. If you flip it over, you see the same thing. So the left-right symmetry of their body results in this symmetric shell in 99% of the cases. In the other 1% of ammonite species, you get these very bizarre non-planar shapes, non-symmetric shapes. So some of these look sort of like other types of seashells that secrete non-planar ones. But you also have these bizarre forms. So the ones here are called nipponites. And for many, many years, I saw these shells and I had no idea. And I really wanted to understand what is driving this very strange shape in these very rare cases. And so that's the question we want to think about. And first of all, we can make some progress by returning to our geometry and realizing that in going from this planar one to this non-planar one, I had two ingredients in the planar ammonite, expand and coil. And I said if I had a twist, then I get this. But that was the key there, was that was a constant twist. And that's what we call a helicospiral shape. On the other hand, if you do the same thing and you have an oscillatory twist, so imagine the animal is twisting one way for a while and then twists back the other way and keeps going like this, you get this shape. And this shape is very reminiscent of this, these shells here, these so-called nipponites. We're going to call those meandering. So returning to the this geometric description, I'm going to do the same sort of thing in the normal and the binormal direction. I'm going to have a growth gradient. I'm going to have a uniform expansion in the normal direction. And then in my tangent, my twist is going to have these three terms, D0, D1, and D2. And so the point is that I can make each of these three shells with the same normal and binormal functions and only changing the type of twist. So in the planospiral case, there's no twist. D0 and D1 are both 0. In this helicospiral case, the D0 is not 0, but D1 is 0. So that's constant twist. And in this meandering case, there's the D0 is 0 and the D1 and D2 are not 0. So we get a oscillatory twist.
So Derek, we have a question from Madhu, I think.
Yes, go ahead, please.
Very quick clarifying question, and you may have said it and I missed it. Self-intersections and how you deal with them in this.
Self-intersections.
Yes.
Yes, it's a good question. I completely ignore it, and it's worth bearing in mind. So because these are very much non-local intersections, right? It's not that the shell is intersecting itself at some previous stage, and you can see even in my picture here that there is some self-intersection there. So bear in mind, I might say more about that later, but in the model, I'm going to completely ignore that.
Got it. Thank you.
Okay, so now I want to think about. So the point of this was that we can get from the normal ammonite to a meandering or a helicospiral non-planar shape simply by changing the way that the animal twists inside the shell. And so the idea that is to think about whether mechanics might be underlying that. So here's our model. Here's the idea of our model. First, the key is that we have to distinguish between growth of the body, which is the soft animal inside the shell, and growth of the shell, which is the secretion coming from the mantle. Okay. And so in my idealization of this process, I'm going to take the animal, the soft animal, to be described by two elastic rods. So that's the red and the blue here. And I'm using 2 elastic rods because, one, it's sort of simpler than trying to model the entire animal. But I think it gives me enough complexity because there's a sort of dorsal and a ventral side of the animal, okay? And so the way I can get a logarithmic spiral is to say that the blue side is growing faster than the red side. And that produces an animal that has a logarithmic spiral shape to it. At each point in time, the animal is secreting, and let's suppose that we highlight the secretion of the shell by the green arrows. Okay, so the red is probably secreting at a slower rate than the blue side because that's how the shell is going to sort of more or less follow the shape of the animal. But our idea is to say, what happens if I have a mismatch between the growth and secretion? So if the secretion doesn't perfectly match the growth, then there's going to be mechanical stress in the body. If the secretion does perfectly match the growth, then the animal will be stress-free and the shell will be the exact same shape as the animal wants to be, right? But we're going to try to ask what happens if we have a mismatch. And the real question mechanically is, could such a stress induce a twist inside the shell? Could this cause the animal to twist purely due to the mechanical forces? And if that does happen, if the animal does twist, then the animal has now rotated the coiling axis, right? So if there's no twist, then there's this axis where there's a gradient in the growth and secretion, and the animal will always be planar, and the shell will always be planar. But if it twists, as you can see in the third picture, that axis of the gradient of secretion has now changed. So even though the animal might still be growing the same way, the shell is not going to look the same. And so that's what we want to probe mathematically. And the way we do that is we say that I have 3 parameters describing the body. And these are really the 3 parameters that just create a logarithmic spiral for the body. And it makes sense to say that the body is taking that sort of logarithmic planar shape, as is the case in 99% of ammonites. And then we have 3 parameters that describe the secretion. And we're thinking of each of these as being fixed, okay, either determined genetically or however. But I'm imagining that these parameters are fixed. And I have a mismatch if the 3 parameters for the secretion are different from the 3 parameters from the body. And then I'm going to say, okay, on top of that, there's some possibility of twisting. Either those D parameters are 0, and that's a planar shell. The D0 is non-zero, and I get a helicospiral shell. Or the D1 is non-zero, and D2 is non-zero, and I get this sort of meandering shell. And so because these are elastic rods, and I'm putting them inside the shell, and they're constrained to be inside the shell, I can write down the mechanical energy of these 2 elastic rods, which have intrinsic curvatures and a particular shell shape that they're stuck in. And therefore, you get a mechanical energy, which becomes just a function of these 3 D parameters, the parameters describing the twist. And from there, we can talk about mechanical energy minimization. And so the idea is I input the body parameters, the secretion parameters, and we also have mechanical stiffness parameters, which really I know almost nothing about, but you'll see how I'm going to at least try to deal with those. And for fixed parameters there, I can compute the energy minimizer for each of the helicospiral and meandering shells. In other words, I have my energy depending only on that one variable. If I'm doing helicospiral, the energy only depends on D0, and I can find the energy minimizer. And I can do the same thing for the meandering shell, where I find which of those parameters minimizes all the possible meandering ones. And then I have 3 different energies. So I have the energy if it didn't twist at all, the energy for constant twist, the lowest constant twist, and the lowest energy meandering shell. And I compare those 3 types, and then simply said the one with the lowest energy is the one we would expect to see. So this is just reiterating the picture. The body itself is always trying to be planar, right? There's no twist built into the body. The twist entirely is coming from whether that has a lower energy than not twisting, okay? So from that, we can do this, I'm calling a morphological phase space. And so in this picture, I am imposing a mismatch. I'm putting all those parameters in. I'm imposing some degree of mismatch between between the body and the secretion. And then my 2 axes here are the ratios of the stiffnesses. So I don't know much about them, but here, let's take a large range of them. And at each point here, I compute the energy minimizing shell, and then I color that point by which one was the lowest. And so right away in this picture, you see that a large part of the space is green, meaning for those mechanical parameters, the planar shell is the energy minimizing one. But interestingly, we do see a small strip of blue, and we see the bottom right corner has red, which means if the mechanical parameters were on those values, then it's actually favorable to secrete this meandering shell or the helicospiral shell. And if we change the degree of stiffness, the degree of mismatch, of course, there's a large parameter space to play with there, but here's a choice of parameters where the blue region has actually become sort of appreciable. Okay, and you can see in this case the planar shell is actually having lots of self-contact. So it's maybe not so surprising that if I change the parameters such that the planar shell is having lots of issues with self-contact, it's also that case where the non-planar shells have become, which have much less self-contact or no self-contact, have become mechanically favorable. So when I first did these calculations, I was thrilled, and my colleague was thrilled just to see some blue. I was sort of going into this thinking green is always going to win, but I was quite excited to see that in some cases blue does win. So if I have a bit of mismatch, stress can induce a twist, and the twist can make the non-planar shell mechanically favorable. But I'm not super satisfied because this is just playing around with a lot of numbers, and they're all very hard to measure because these animals have been extinct for a very long time. So is there a way to be quantitative? Well, I mean, that's an interesting question on its own. I'm studying extinct animals just by looking at their fossils, but I want to at least think about being quantitative and making something along the lines of a prediction. And so here's what we're going to do. I'm going to fix a small degree of mismatch. In fact, only in one of those parameters. So the coiling looks the same. They're sort of trying to produce the same logarithmic spiral, the body and the secretion. It's just that the body is growing a bit faster than it's secreting. And so it's becoming compressed. And then I still don't know much about the mechanical parameters, but as I change my coiling parameters, what I can do is I can sweep through this parameter space of the mechanics. In other words, create an entire diagram like this, and just count how many greens, how many blues, how many reds there were. And then I'll assign to that coiling parameters whichever bin got the most. So if green was the most likely, or if most of the bins were green, then I'll say that green is most likely for those choice of parameters. And so if you do that, so now I get this two-dimensional space on the coiling parameters, coiling gradient and the expansion rate. And again, you get bits of each one winning. So over on the right, I'm coloring these points RGB values based on what percent the different types were the energy minimizer. And you do see a region in the middle where the meandering shell is winning more of the mechanical space than the others. And over on the right, the planar shell is by far winning the most. And just to give you, make that to try to move this thing again. Sorry. If I decrease the amount of mismatch, so this is like a 2.5% compression or extension versus 7.5%, you see that this feature goes away. So in this case, the green wins everything, and it wants to stay planar. So having done that, what we can now do is take lots of these non-planar ammonite shells, and for each one of these, I sort of fit my model, my geometry, to the image of the shell, which is a way for me to extract the coiling parameters of those shells. Okay, so I did this for something like 15 to 20 of the helicospiral and the meandering nipponite shells, and in that way we can put those shells on this diagram. And so that's what the X's are here. The red X's are the helicospiral ones we sampled, and the blues are the meandering. And I mean, it's sort of roughly agreeing with what our model is predicted. So the blue ones are pretty much all living in the range where the model said blue is going to be mechanically favorable. The reds are mostly living to the left where helicospiral is favorable. And none of these shells are having coiling parameters over on the right where the model says that those are very mechanically unfavorable. So that's at the very least consistent. And just to note one more interesting shell. So this guy is called Didymoceras. These are very rare, quite beautiful shells. The best way to characterize these shells is to say that they are doing each of these different types of coiling at different points in their development. So as a juvenile, they start out with a planar spiral shape, and then in the sort of middle stage, the red, they're doing the sort of helicospiral shape, and then at the end, they go back to being planar spiral. But notice that the plane in which they're coiling at the end has been rotated from the plane in which it started. And that can only happen if there was a sort of meandering transition somewhere in there. And so we can simulate these guys by simply saying that, well, rather than have these coiling and secretion, growth and secretion parameters being fixed throughout the lifetime of the animal, suppose that those parameters change during development. If those parameters are changing during development, then at different points, meandering might be favorable and helicospiral might be favorable. And so there's a sort of consistent idea there that I can produce this shape via a varying parameters during development, but in particular, you notice that if you looked at those morpho spaces I was creating, the blue was always living in between the red and the green. In other words, if my parameters are going to kind of somehow transition to go from planar spiral to helicospiral, the model suggests that you can only do that if you sort of pass through the meandering stage, which is quite interesting that that's what these shells seem to do. Okay, I'm going to leave the coiling now and look at another feature of ammonites, which is the ribbing. So back to this picture. So forgetting the planar or non-planar shape, what you notice in this picture is that the shells, for the most part, are not smooth, but they're characterized by these ribs. Okay, there's a few shells sort of up in the corner that are quite close to smooth or smooth, but most of these species have clear ribbing pattern. If you go back to the 70s, actually earlier than that, 1967, David Rock was working on shells, and he created what is usually thought of as the first theoretical morphospace. So what he did was he was studying ammonites, and he looked at the cross section. So imagine slicing open the ammonite, and you can measure these different things, so sort of how far apart are the different edges. And from those, you can make these different parameters that he called W and D, and then you get this W and D morphospace. I'm not too worried about the specifics of what W and D represents, but you have these pictures on here, and what it shows you is that it's a way to characterize the type of coiling of the different ammonites. And so effectively all the ammonites would live, the planar ones would live somewhere on this diagram. All of the coloring and the lines and the arrows, they represent different things about evolutionary trends that have been observed or where different species have sort of been found on this chart. What is particularly interesting, what I want to focus on, is the correlation between location on this chart and ribbing. And what you find is that ammonites that live sort of down in this bottom left, which are characterized by a very rapid expansion rate, tend to be very smooth. And related to those is the nautilus, as I said, the living cousin of the ammonite. Nautilus has that same geometry and is also a very smooth shell. If you go to the upper right of the diagram, you get these shells that look almost like a rope that you've just coiled up. So the expansion rate is very small on these shells, and those shells tend to have very dense ribbing. And then somewhere in the middle, you get these kind of intermediate values that have sort of like, they have a ribbing, but it's more of a coarse ribbing. Okay, so this is a trend that is very well conserved. And what we want to try to do is try to understand, well, why there's this difference in ribbing, but why is there ribbing in the first place? And so returning to the idea of how the shell grows, I want to look at this a little bit closer. So what happens is, here's a close-up of the mantle and the shell in yellow. The mantle has a sort of interesting three-pronged structure. It has these three lobes. And what happens is there's this so-called periostracum, which is secreted between the outer and the middle lobe. And that periostracum, which is itself an elastic material, but very, very thin, is secreted there and then sort of folds around the edge of the shell and forms a sort of almost a skin for the shell. And that kind of, well, you can think that forms a protective layer for calcification of new shell material, which is the sort of blue color in there. If you think about the physics of that, so what physical forces are in that? Well, you have this mantle periostracum combination, which is a bit complicated on its own because the mantle is a soft three-lobed material. The periostracum is a very thin elastic material. These are attaching to the current shell. Okay, so that's going to induce mechanical forces. But they're also attaching to the shell, which has an orientation. And so in particular, the shell has an angle in what's called the generative zone. So imagine that the shell was just a cone. If that cone had a particular angle, then there's going to be some resistance to changing that angle because because this generative zone is itself made of material that has a resistance to change. So we built a very simple mechanical model of this process. And now, whereas in the previous model, I was kind of thinking of the entire body of the ammonite, the mollusk inside the shell. In this case, I'm going to ignore that. I'm only going to focus on the mechanics of the mantle edge where the actual secretion is taking place. And so I'm going to think of the mantle edge as being an elastic ring. Well, actually, it's the mantle edge and the periostracum forming a sort of bi-ring, but you can kind of work out that details. And in the end, it's effectively a single ring, which is the red curve. And in my model, so I'm also going to imagine unwrapping the shell. So I'm going to ignore the fact that there's coiling and the secretion is not happening the same on the dorsal and the ventral side. And let's just imagine it's trying to construct a cone. So the calcified bit of the shell is already there, and that has some angle, which is phi, which I said that's the sort of angle of the current generative zone. And the current mantle is a ring that has some radius that it wants to be, sort of stress-free radius, which is capital R. And so you can write down the geometry is straightforward enough. S is the radius that the mantle is going to take in its equilibrium state. So capital R is the state when it's dissected, and S is the under the force of being attached to the shell is sort of the current radius. The mechanics is really just a force balance for an elastic ring. Notice that there's this extra force d phi dz. That's the sort of torque spring force of the generative zone resisting a change in its current angle. And the growth, so whatever the mantle is doing, it's trying to grow, is captured in this function R of Z. So the idea is that if you input some rule for how it's growing and you input the material parameters, then you would solve this set of equations for the shell pattern, which is the function S of Z. Now we can simplify things. So suppose that the growth is linear. So the radius of the stress-free mantle ring, suppose that's just some scalar times Z, Z being the arc length following the center line of the shell. Then we get a simplified system as appearing here. We can take that further and suppose that we suppose that S is close to R. So this is like if we don't deviate very much from the stress-free state, then we can solve this system asymptotically and we get this exact solution right here. So here X is my perturbation of S from being a cone, and you get this exact solution like this, where the two free parameters are simply an expansion rate and an elastic parameter. And as you would expect, it's doing this oscillating thing with increasing amplitude and also an increasing period. And you can kind of intuitively make sense of what's going on here. So these sort of 4 points, as the shell is increasing in its radius, the mantle is increasing in tension, and at some point is going to have enough tension to sort of pull the radius back in, but there's a sort of momentum to it because of this resistance to change in angle, and so it overshoots, and then it becomes in compression, and now the mantle is in increasing compression when we're in the valleys, and then the compression becomes high enough that it goes back out again, and so you get this oscillating pattern. Now again, that's nice. We can produce something that has oscillations. The model, kind of is based on our physical intuition for what we think is going on. But again, it's not very satisfying unless we can at least try to be quantitative in some way. And so the way we're going to do that in this case is we can take a shell and fit a logarithmic spiral to the outer edge of the shell, the inner edge of the shell, and the center line. And then you can write each of those as logarithmic spirals. And then from there, just simple geometry, you can work out the arc length, which is the parameter Z in the model. And so the idea would be this. I do that for a shell, and by extracting those best fit logarithmic spirals, what I'm really doing is I'm measuring the expansion rate. And so I'm sort of unwrapping those spirals, and I'm just thinking of the shell as a cone. And then I feed that expansion rate into my model, and I'm going to output some ribbing pattern, some oscillations, which I can then put back on the shell. And of course, there is the mechanical parameter, which I have no idea about, but I can at least find a sort of ballpark based on Young's modulus of nautilus tissue and things like that. But we also sort of think of that as a single fitting parameter. And well, you can see it sort of does a quite reasonable job. So the ribbing blue is quite nicely overlaying with the ribbing we see on the actual shell. We can take that a little bit further. So two things to observe. This was my exact solution. The ribbing amplitude has this Z to the 1/2. And so what that means is that I expect the amplitude to be growing as the square root of the arc length of the center line. And we can measure all of that on the shell. So I take these shell images, I measure an amplitude by taking these radial lines and take the difference from peak to valley. And then I plot that amplitude as a function of arc length. And if I plot that on a log-log plot, I get these scattered dots. But they are quite closely following this line, which has a slope 1/2. And 1/2 is what we expected from the linearization of the model. Also worth noting, I guess, that this is a fossil that has been around for a very long time. So we kind of expect some degradation. So the noisy amplitude is not surprising, but I was sort of pleasantly surprised that this is roughly following the trend. Inside the cosine, we have this omega log Z, which is telling us something about the spacing of the peaks. And that's another thing that we can compare to a quantitative measurement. So what I did was I took something like 20 different specimens of the same species of ammonite, and for a fixed mechanical parameter, which I only fit once, I measure how many spines or how many ribs there are on the outside and how many ribs there are on the inside as I follow around the shell. And then I measure the expansion rate for that shell, and the expansion rate is living in that parameter omega. And this is the data is the blue and the green, and the curves are the model. And again, it's doing quite a reasonable job, I'd say. And there's taking one of those points and putting that shape back on the shell. Yeah, I mean, it's probably worth pointing out, if you haven't already observed, that this is I'm really cheating in the model, because here I'm treating the outside of the shell and the inside, the ventral and dorsal sides, sort of separately. But of course, my model doesn't do that. My model actually treats them as a single cone. The model is ignoring the fact that they're curved. What you can see is happening in the real shell is you have these bifurcations. So these two spines, if you can see my mouse, or these two ribs here, are bifurcating from this single rib that's appearing on the inside. So the model is definitely inaccurate in that regard. But nevertheless, it's a little bit interesting, I think, that we still do quite a good job of capturing the sort of total number of ribs just as a function of arc length via this simple formula. One thing we can do, well, so that formula is a cosine, which is very smooth ribbing. Actually, most ammonites are not perfectly smooth, but actually characterized by peaks that are sharp and valleys that are sort of long and smooth. But we can imagine adapting the model in such a way that if my elastic parameter is bimodal, so really if it just anisotropically responds to tension differently than compression, which is kind of characteristic of a number of biological tissues. In other words, if it responds more strongly to being in tension than in compression, then conceptually you imagine that you spend less time in the peaks when this tissue is in tension than in the valleys when it's in compression. And you can play with different kind of parameters there and get these different profiles, which do a sort of reasonable job of capturing some of the profiles we actually observe on ammonite shells. Going back to this morphospace and the question I started with, what you find in the model is that, in fact, the expansion rate plays a really big role in how the pattern sort of evolves. In particular, if I take the exact same initial conditions, the exact same mechanical parameter, and only change the expansion rate, this is what you get. You get exactly what is observed, which is that if I have a really large expansion rate, effectively the oscillations are being sort of washed away as the thing is just growing too quickly. The oscillations comparatively become very small. And so in this simulation down here of something resembling a nautilus, it is starting with oscillations. I'm inputting the oscillations into the system, but because it's growing so quickly, at some point it just looks very smooth. Whereas if it's growing very slowly, those oscillations persist and you get this very dense pattern. And so we do seem to reproduce this observation of the morphospace. Right, I think I'll stop there. So just a quick summary. We have looked at a couple of features of ammonites, and I think one of the really interesting things is just the idea that we have in the case of the meandering and the helicospiral shell, this is a symmetric animal that is secreting an asymmetric shell entirely because there's a mechanical influence. And the key ingredient there is mismatch. If you have some mismatch between the growth of the animal and the secretion of the shell, you get these mechanical forces which can account for these really interesting patterns. Mismatch in a different way, thinking in the second part, the ribbing is also coming from a mismatch between the shell and the animal, but this is now between the sort of radius, sort of orthogonal direction, the radius of the shell and the radius of the last sort of leading edge of the mantle. And this gives us a nice way to understand why the nautilus is so smooth. Even if it had ribs, we would still effectively see a smooth shell. Lots of stuff that we're still curious about and either working on or would hope to work on. One is this is very much focused at tissue scale mechanics and the pattern formation that comes from that. It'd be really nice to link that to some of the things that are underlying sort of where's the connection with chemical patterns and genetic and molecular scale as well. And lots of other patterns that I don't have time to talk about today, but hopefully some of the pictures earlier give you appreciation of the beautiful and diverse patterns that still, many of which still need to be explained. And so I will end there. I thank my colleagues, Alain Gorielli and Rajesh Shirat, who really the three of us for the past, I say almost 10 years have really been in love with trying to understand different features of shells. And I thank you for your attention.
Great, thanks so much, Professor Moulton. Anyone have any questions? I actually have one to start us off. Henry has raised his hand. Excellent, Henry.
No, go ahead. You first.
Okay. I was just curious about individual variation within species and how much repeatability there is, I guess, versus sort of behavior that could be shaping these things, given that the animal's twisting around. I mean, can they choose to be doing this, or is this sort of like a set thing? That makes sense.
So, I mean, the twisting, our view of the twisting is that this isn't, I mean, this always interested me, because you can work out the geometry, and for a long time we say, okay, in fact, not before us, it was clear that to make this shape, there's a rotation inside. And so why is this thing rotating? Is it choosing to rotate? That's kind of a weird thing to say. Is it choosing to rotate inside its shell? And it has to do it in a very specific way to make this pattern. So to me, I think it's a much cleaner explanation to say it's not thinking about, it's just doing whatever it does. It's looking for food, and because mechanical forces are acting on it, they're taking care of it and causing it to rotate in particular ways. Variability within a species, there is certainly some, but much smaller. So I mean, from a sort of parametric point of view, yeah, you'll see some variation. I don't know if I can. Some of these I've forgotten whether some of them are all from the same species, but yeah, you'll see some variation, but yes, very little. I mean, so and these species that produce the non-planar ones, the entire species will do that. I should clarify that. So it's not that some individuals might make a non-planar shell, whereas their cousin is making a normal shell, a planar one. It's the case that there are species that create the non-planar ones, and our understanding would be that they have, for whatever genetic mutation, adopted this mismatch in their coiling or their secretion, and that has then been conserved across that species, so that they're all feeling that mechanical force that causes them to do this non-constant or otherwise twisting.
Gotcha. Thank you so much. Henry.
All right, so mine actually is sort of similar, but sort of few taxonomic levels up, which is you've got these mollusk shells and you have a handful of parameters that can explain a lot of the shape. How well, or do you know how well these parameters actually match onto proposed species delineations and phylogenetic relationships? Like, are there any surprises where it's like, oh, you can go from this to this with this massive change by tweaking this one parameter a little bit, and so maybe these species are more closely related than their external shell shape would suggest, sort of thing? I don't know if I'm making that clear.
I think it's a fantastic question. I don't think we have got much of an answer yet. I mean, I can play with parameters and I can say changing this one has more of an influence on the shape than you see than changing this one. I think what it really needs is to connect this. I think it really comes to like the multi-scale idea. So what is this parameter? I you know, at the tissue scale, I think to make clearer connections, I think we need to have an understanding of how that is it that it's eating more, for instance, right. and that's feeding into this parameter, and all you need to do is eat a little bit more so that you can you grow a little bit faster, and then you see this change in form. And right, yeah. We kind of are getting close where we can push in that direction. I have a different collaboration with a cell biologist and we are sort of measuring things on living specimens and their growing shells. We're kind of pushing in that direction, but I don't think we have so much to say yet. But it's a fantastic thing to think about.
Actually, the collaboration you mentioned sort of leads into a second question I had, which is, are there, and can your system sort of account for, are there poisons or chemicals that induce certain types of shell deformities in the mollusks by essentially changing some of these terms or impeding mechanosensation or something like that?
Yeah. I think mollusks for the most part haven't been studied so much in that regard. I mean, so one example that comes to mind, So they were trying to understand coiling in gastropods.
Right.
So why does a snail coil?
Mhm.
You know, so snails. I mean, this was a separate part of our analysis that I didn't fit into this talk. But gastropods, like the snails you find in your yard, they are almost never planar. To say, okay, well, what is it about them that's causing them to be non-planar? They must be twisting. Of course, their body plan is not symmetric, so there's a difference there.
Right.
But there was some genetic studies on trying to change that. So you can early on do a knockout that makes a particular snail, lose its chirality, and therefore it doesn't secrete a non-planar shell anymore.
Mm-hmm.
Yeah, I don't know of two other examples where there's a clear link that's been established between doing something chemically and having a large change on the shell form. But it's definitely an interesting direction to go.
Cool. Thanks. Really cool talk.
Thank you.
Raghavan, please.
Yes, and this was really interesting. Actually, it popped up a lot of questions in my mind, but just a couple of them that I have. So in the morpho space that you showed at last, so between Nautilus and the top one, so what happens if there is an oscillation in the growth rate? So let's say the organism grows faster for some time, so that it generates a smoother profile, and then it slows down. So then, how would the ribbing be? Is it the one in the center, or is it different?
So the growth rate is surprisingly constant. But I should clarify that. So what I mean is that those the ones in that morpho space, it's assumed that the growth rate is staying the same throughout their development. But by growth rate, what I really mean is when they're secreting the shell, they are doing it in the same way. You know, when I go back to my geometry, my growth vectors are staying constant, very close to constant throughout development. What's interesting is that doesn't mean that they're always growing at the same rate as in sort of in an absolute time. So they might go a period, you know, it's cold, they're not finding enough food, they're not growing the shell as quickly, and then there's a lot of food present and they're growing the shell more quickly. What's interesting is, and this, I don't have an answer to this, but I think it's a really good question also, which is with that kind of variability, they nevertheless are secreting in a way that looks constant. So if I ignore sort of absolute time and just take my parameter to be, say, the arc length of the shell, things look constant in that parameter. So there's a really kind of remarkable robustness to the way that they grow. In principle, we can play with that. We can say, suppose that they actually are not doing something robust, but for some period their expansion rate, you know, increases quite rapidly and then slows down again. You're going to get a kind of funny looking shell, but it's not going to be. It's going to sort of lose its mathematical regularity. And as far as the oscillations, I don't know. Yeah, you'd be somewhere in between where it'd be kind of smoothed out, but then maybe it would come back. But like I said, there was this Didymosurus shell I showed. And the idea there, I think very much is that the sort of growth parameters change from juvenile to middle stage and so on.
Yeah, thanks. So I have one more question. So another thing that came to my mind is, this is a bit out of context, but you see actually kind of similar folding mechanisms during heart development. I know now I'm talking like, as Henry said, like several taxonomical levels, like completely unrelated. But during heart folding, during the formation of the heart, it's like an elastic tube that actually experiences two asymmetrical forces and starts folding in opposite directions. And then there's like a couple of helical twistings, and then the heart formation goes on. So my question was like, do you think this model can be extrapolated to such complex organizations?
Yeah, I mean, it's interesting. I was earlier this year presenting the work on the coiling, not the ribbing, in a conference on asymmetry. And it was the entire focus of the conference was where does asymmetry come from? And someone was talking about that exact problem, the heart tube. And actually they were in fact working on this as a mechanical instability kind of idea.
Mm.
So there is some work already in that kind of regard, and definitely that's a generic interest of our group, which is to understand symmetry breaking and formation of chiral structures from a mechanical point of view.
Yeah, great. Thanks very much.
Thank you.
Great, thank you. We have one other question in the chat. Nate asks, Nate says, I was curious if the smoothing of the ribbing due to the growth rate can be attributed to environmental conditions similar to tree rings changing due to their yearly environment. What are your thoughts? Thank you for your time.
So one answer is I kind of comes back to what I was saying, that I think the so growth rate is going to be connected to the environment, but it seems that the actual growth rate that I'm talking about seems sort of environmentally independent in some amazing way. So my guess is that I don't know about the smoothing being connected to environment. I can comment quickly on another example where environment really is doing something significant. So we were studying a shell that is found in Japan that makes these spines. Let's see if I have. I don't have that one, but here's a sort of similar one. I don't know if you can see that. So it makes these sort of spine patterns, which again, we in our view, that's emerging due to a mechanical instability, that the mantle is growing too fast for the edge, and it does like a buckling, which gets cemented in place. And this species, if you took them from one environment to another, so if you found them on the beach or in the ocean, and then you moved them into an aquarium, they stopped making those spines. So I talk about robustness. There seems to be a robustness for some features, but not all features, and that's one feature where the environment really definitely is playing a strong role. But yeah, I don't know of examples where there's sort of a yearly thing that it is appearing in the shell. They're not really growing in such a way. They say, you point to that bit of the shell and say, okay, that happened in summer and that happened in spring. I don't think they are normally growing in quite that way, but they are some interesting examples of being impacted by the environment.
Gotcha. Thank you so much. Any other last questions?
Well, thank you so much for
Yeah, thank you. Thank you for taking the time to talk to us and for that fascinating talk. It was really very enjoyable. Oh, that's just a wave. Okay, sounds great. Thank you so much, Professor Moulton, for joining us.
Okay, well, thank you.
Thanks
Have
everyone
a good
else.